Welcome viewers. Today in this session we will be talking about restriction enzymes. We will be covering about what are restriction enzymes, why are they found in bacteria, and mainly the different types of restriction enzymes found in bacteria. Before we move into all of these, first let us know in brief what are restriction enzymes. Restriction enzymes are a subset of nuclease. Nuclease are those kind of enzymes which are able to cleave the nucleic acids at specific or at random points. Restriction endonuclease are a type of endonuclease which means it is able to cleave the DNA only after binding or recognizing certain target sequences. Like shown in this diagram, we can see that the restriction enzymes are a part of restriction modification system. Now let us move to what is restriction modification system. So the restriction is the DNA which is containing certain recognition sequences is cleaved by a enzyme after recognizing this sequence. As a result, it produces double-stranded breaks in the DNA molecule. On the other hand, the modification is catalyzed by the presence of certain methyl transferase enzyme which add methyl groups to the specific bases present in the recognition sequence. So, the restriction endonuclease and the methylase transferase are together constitute the restriction modification system of bacteria. The study of restriction modification system helps us to understand or to know better about the bacterial immune response. It helps the bacteria in distinguishing the self DNA from non-self DNA. How it is accomplished? Let us understand this with the help of another example or with the help of another diagram. As shown in this figure, we can see that the DNA which is containing this kind of specific bases in them is methylated by a methyl transferase enzyme. Now, since it is methylated, the restriction endonuclease, though it is synthesized within the bacterial cell, is not able to cleave its own DNA because of the presence of methyl group. As a result, the bacterial DNA is protected from its own endonuclease activity. While if there is any viral attack in it, then what will happen? Due to the viral attack, the bacteria is able to distinguish the self DNA from non-self DNA. As a result, the presence of restriction endonuclease in them cleaves the viral DNA, whereas it has got no effect on its own DNA due to the presence of methyl group. In this way, the restriction enzymes are able to protect its own DNA, whereas it is able to cleave or digest the foreign DNA material. So, let us now come to the functions of restriction enzyme. The function of restriction enzyme are twofold. It protects its own DNA by methylating the bases or certain bases present in the recognition sequence. The second important function of restriction enzyme is that it has the ability to digest the foreign DNA material.
Ma'am, does it mean that bacteria is always resistant to bacteriophage attack?
No, always the bacteria cannot overcome the bacteriophage attack. It is due to the synthesis of hydroxymethyl transferase produced in certain bacteriophages like T4. Therefore, this kind of phages attack the bacterial cell. The bacteria is not able to resist their attack because the basis in the viral DNA is already methylated. As a result, the restriction endonuclease present within the bacterial cell is not able to cleave the viral DNA and the virus is able to invade the bacterial cell. Now let us move to the different types of restriction enzymes. The restriction enzymes can be divided into different types based on its subunits or the type of the subunits, cofactor requirements and the enzymatic cleavage mechanism. So based on these, the restriction enzymes have been broadly divided into type 1, type 2, type 3 and type 4. The restriction enzymes or these 4 different types of restriction enzymes are prevalent in common bacteria like Escherichia coli, Bacillus subtilis, Haemophilus influenzae, etc. Now let us move to the type 1 restriction enzyme. The type 1 restriction enzyme is a multifunctional and it is a complex enzyme. If we look at the subunits of type 1 restriction enzyme, it is made up of 3 different subunits. One is the methylase subunit, which is denoted by the letter M, the specificity subunit denoted by the letter S, and the restriction subunit denoted by the letter R. Now, all of these 3 subunits assemble together to form a heteromeric holoenzyme. The characteristic feature of type 1 restriction enzyme is that it is able to catalyze both restriction and modification by the same enzyme. The gene which is coding for the methylase activity is denoted as HSDM, for restriction activity as HSDR, and for specificity HSDS, where HSD stands for host specificity for DNA. So, after looking at the subunits, we can see that this is a heteromeric type 1 enzyme, the example of which is ECO-K1, and the 3 subunits assemble together to form the complete holoenzyme. Now, let us look into the mechanism of cleavage of type 1. The recognition site of type 1 is an asymmetrical DNA sequence. It can be divided into 2 domains of specific region. One region which is 3 to 4 base pairs long, another region which is 4 to 5 base pairs long, and both the regions are separated by a variable non-specific region of 4 to 8 base pairs long. So, all of these 3 features together constitute the recognition site for type 1. The mode of action of type 1 restriction enzyme depends upon the methylated state of the target sequence. If the target sequence is unmethylated, then only the type 1 restriction enzyme is able to restrict the DNA or is able to cleave the DNA. If the target sequence is hemimethylated, then the type 1 restriction enzymes act only as a maintenance methyltransferase enzyme and methylates the other strand. If the target sequence is fully methylated, then it is not able to undergo any kind of enzymatic reaction and the type 1 restriction enzyme dissociates from the methylated sequence. Now let us move to the mechanism of how the type 1 restriction enzyme performs cleavage, or how the cleavage is performed by type 1 restriction enzyme. We will explain or see the proposed model for cleavage. So, as shown in this diagram, we can see the type 1 restriction enzyme containing a specific unit along with its restriction units being or recognizing a non-methylated target sequence. After it binds to a non-methylated target sequence, it translocates DNA towards itself by forming loops and with the hydrolysis or with the help of hydrolysis of ATP. Thus, the DNA translocase activity is an energy-driven reaction catalyzed by hydrolysis of ATP. The DNA cleavage occurs at a point when there is a collision with a type 1 restriction enzyme colliding from another site or with an unusual DNA structure. The type 1 restriction enzyme is able to cleave the DNA only after a certain base pair or only after 1000 base pair from the recognition site. So thus the exact location of cleavage of type 1 restriction enzyme cannot be certain. It can only cleave the DNA when it collides from another structure with another structure or with another type 1. So in this way we can see how the cleavage mechanism, or we can understand better the cleavage mechanism by type 1 restriction enzyme. Thus we can say that the type 1 restriction enzyme is a DNA methyltransferase, is a restriction endonuclease, is a DNA dependent ATPase and a DNA translocase.
Ma'am, I have a question. What is the function of the translocase?
With the help of translocase domain, which is present in the restriction domain of type 1 subunit or type 1 enzyme, it can form DNA loops and it is helped to pull the DNA towards the enzyme. So with the help of translocase, the DNA, or rather the enzyme, translocates over the DNA until and unless it meets with another restriction enzyme translocating from another site. So in this way, it helps it to scan the DNA molecule. So we can see that the translocase property of type 1 restriction enzymes enables it to cleave the DNA molecule. So let us schematically see what are the characteristic features of type 1. So the recognition sequence is bipartite interrupted. The subunit structure of type 1 is a heteromeric composed of 2R, that is 2 restriction subunits, 2 methylase subunits and 1 specificity subunit. It is activated by the presence of magnesium ion, ATP and ADO-MET. And as well as the example of type 1 restriction enzyme is ECO-K1.
Ma'am, why are type 1 restriction enzymes not used in recombinant DNA technology?
The type 1 restriction enzymes are not used in recombinant DNA technology because of two main basic facts. The first is the restriction site for type 1 enzyme is non-symmetrical or asymmetrical in nature, and the molecule or the enzyme cuts the DNA at variable position from the restriction site. As a result, the exact cleavage site of type 1 restriction enzyme cannot be certain. The second important point is both the modification, that is a methylation, and restriction enzyme activities of type 1 are carried out by the same enzyme. Hence, the recognition site may be modified by the methylase before it is cut by the restriction site of the same enzyme. So in this way, it does not help us to use it commercially in terms of generating clones through recombinant DNA technology. Type 2 restriction enzymes are much more simpler in nature, and they are mainly composed of two units. That is, the modification and the restriction activities are carried out by two different proteins which recognize the same target sequence. Hence, the recognition site is not dependent on methylase and it can be used separately for cleaving the DNA molecule. The restriction enzyme activities requires the presence of magnesium ions only. It does not require the presence of ATP, and hence the type 2 restriction enzyme are ATP independent enzymes. While for methylation activity carried by type 2, they require the presence of ADO-MET. Why? Because the ADO-MET is the donor of methyl group for the methyltransferase enzyme activity. Now, as we can see in this diagram, the type 2 restriction enzyme is a monomer of a restriction and a specificity subunit, or it can be a homodimer in other types of type 2 restriction enzyme. So, it is able to cleave the DNA at certain basic points with the help of magnesium ions, which we will be covering later. Now, let us see or study the different subtypes of type 2 restriction enzyme. The first main type is type 2P, an example of which is EcoR5. The type 2 enzyme is a subset of type 2 restriction enzyme, an example of which is EcoR5, and it is able to bind to palindromic DNA sequence. Now, it generally binds to the palindromic sequence in the form of dimer. One active subunit binding on one strand, while the other active subunit binding on the complementary strand. In this way, they are able to cleave DNA sequences within the recognition site. The next example or the next subtype of type 2 is type 2S. The characteristic feature is the restriction enzyme binds to a symmetrical DNA sequence and cleaves DNA 9 base pairs away from the restriction site on the top strand and 13 base pairs away from the other complementary strand. So, the example of which is FOC1. So, the type 2P and type 2S, the diagram of which is shown here. Now, there are the other three basic types. One is type 2F, type 2B and type 2E. In type 2B, the restriction enzyme or the type 2B restriction enzyme makes double-stranded DNA breaks on either side of the recognition site. Thereby, you are yielding a fragment which is containing the intact recognition site. The example of which is BCG1. The characteristic feature of type 2E is very interesting. Why? Because here the type 2E is characterized not by the reaction site, not by the nature of reaction mechanism, but by the nature of recognition site. That is, here the recognition site is present in two copies. Now, one of the copies of the recognition site is acting as an enhancer for the cleavage mechanism of the first copy, and the second copy remains uncut by the enzyme. So, here the second copy is acting as an allosteric modulator, or it is acting as an enhancer for the type 2E enzyme to cleave the first copy of the target sequence. The example of which is NAE1. The other last type or the subtype is type 2F. In type 2F, again the recognition sequence is present in two copies, but it is slightly different from the type 2E in that the restriction site or the enzyme cleaves both the restriction site, and here there is no effect of enhancers. So, the difference between type 2F from type 2E is that the restriction enzyme is cleaving both the restriction or the target sequences in contrast to that of type 2E. So, in this way we can see that the type 2 can be divided into 5 main subtypes. Now, let us move to the mechanism of cleavage by type 2 restriction enzymes. Now, generally the restriction enzyme or any enzyme we know contains an active site. The active site or the amino acid sequences present in the active site is responsible for the mechanism of cleavage. Now, in case of your restriction enzyme cleavage, the restriction enzyme first binds to a DNA strand. The binding of enzyme to the DNA is non-specific in nature. Why non-specific? It can bind randomly at any site on the DNA molecule. After it becomes bound to the surface of DNA molecule, the restriction enzyme gets a conformational change. The conformational change helps in adapting the protein strongly to the DNA molecule. After this, the enzyme slides over the DNA molecule in search of its target sequence with the help of linear diffusion or facilitated diffusion. After it encounters a specific target sequence, the enzyme adheres to the DNA with the help of hydrogen bonds. The hydrogen bonds helps in the tight association of the enzyme with the bases present in the target sequence. During this process, more hydrogen bonds are formed and water molecules and ions are released so that the association between the DNA and the enzyme becomes more tight. The cleavage is done or achieved when the magnesium ion is present. The magnesium ion helps in the cleavage of your DNA molecule at certain restriction or certain sites present within the restriction or recognition sequence.
Ma'am, what is the role of the magnesium ions?
The role of magnesium ion is that it forms a complex with the enzyme or with the amino acids present in the active site, and it helps in the catalysis of the cleavage reaction carried out by restriction endonuclease. So the mechanism is a bit complicated and it's not been covered, but for till now you can know that the magnesium ion is involved in the catalysis or in the hydrolysis of phosphodiester bond, which is resulting in the break on the DNA molecule. Now let us move to the fact that why type II restriction enzyme is commonly used in recombinant DNA technology. Two main reasons. One reason is that since the modification and the restriction endonuclease activity are carried by two different enzymes, and therefore as a result the restriction endonuclease enzyme can be used without modifying the target DNA. Whereas in type 1, both the activities, restriction and modification, were carried out by the same enzyme. The second important point is we exactly know where the type II restriction enzyme is going to make a cut on the DNA molecule. As a result, the target sequence, since it is known, so we can manipulate or synthesize the DNA or the strands accordingly in order to enable it to get cut by the required enzyme. The third important point is the reaction carried out by the endonuclease is very simple and only requires magnesium ions and no other cofactors. So all these features makes type II more attractive towards recombinant DNA technology. Let us now move to the type III restriction enzyme. The type III restriction enzyme was first discovered in Escherichia coli strain K12 harboring P1 lysogen. They have the characteristic feature very similar to type I restriction enzyme, and they also cleave DNA 25 to 27 base pairs away from the restriction site. They also can carry out restriction and modification system similar to type I and which are both performed by the same enzyme. Let us now see about the last class of restriction enzyme, that is type IV. The type IV restriction enzyme is different from the others in that it is able to cleave the modified bases. The recognition site for type IV restriction enzyme is RMC, where R stands for any kind of purine, and the C stands for cytosine. Now the restriction enzyme of type IV was first found in Escherichia coli, and it was encoded by the genes MCRBC, MRR, MCRA genes. Now it can bind to the DNA only in the presence of GTP. With the hydrolysis of GTP, it is able to cleave the DNA molecule. So this is the characteristic feature of type IV.